So it's my pleasure to introduce our speaker, Klaus Gramann from the Biological Psychology and Neuroergonomics Institute. Well, it's Institute for Psychology and Ergonomics at Berlin Institute of Technology. Klaus Gramann received his PhD in psychology from, is RWTH short for something, Klaus?
Yeah, that's the Rheinisch-Westfälisch-Technische Hochschule Aachen.
Okay, I'm glad you said it then. Not in Germany. As a postdoc with the LMU Munich, Germany, and the Schwartz Center for Computational Neuroscience at UCSD. After working as a visiting professor at the National Chiao Tung University in Taiwan, he became the chair of biopsychology and neuroergonomics with the Technical University of Berlin in Germany. Was a professor with the University of Technology Sydney in Australia, and is an international scholar at the University of California, San Diego currently. So his research covers neural foundations of cognitive processes with a focus on brain dynamics of embodied cognitive processes, and he directs the Berlin Mobile Brain Body Imaging Labs, B-Mobile, that focuses on imaging human brain dynamics in actively behaving participants. So look forward to hearing more. And with that, you can take it away. Thank you.
Thank you very much. Thank you for the kind introduction and for the invitation. And I hope to be able to come by in person very soon. So I'm sorry that I couldn't be there. But I'm very happy to present our work to your group because I think we have like significant overlap. And I hope I can show you some interesting research that we do in Berlin. So I'm going to talk about mobile brain-body imaging as a tool to investigate the neural basis of embodied cognition. And I don't have to bring owls to Athens. I think most of you are aware that brain dynamics change dependent on the behavioral state. That has been shown in animal research for decades now, and it becomes clearer in human research too. And as Weir and Dickinson stated, that the neural architecture that is the hardwire, the network, remains relatively static, that the functional connectivity, the changes of activity, the hapness in brain networks changes almost instantaneously according to the behavioral context. And they showed that in the fruit fly, single cell recordings in the fan-shaped body of the fruit fly, where single neuron activity drastically changes the moment that the fruit fly is airborne as compared to resting on an air-puff mouse ball, basically. And this is a brain region in the fruit fly that is not directly related to motor control or flight or visual input, but some mediating area. And this is important as this has been observed in rodents and in many species, but not very often in human participants, because the traditional brain imaging approaches that we're using for decades now require participants to lie supine in a scanner or to sit in dark experimental lab spaces, being forced to focus on the screen and respond to predefined rules in an experiment. And we record these millions of bits of brain dynamics in these imaging modalities, and then we compare that to a very reduced behavioral amount of information that is one bit of a button press at the end of the trial according to pre-specified rules. And that arguably is not what the human brain has evolved to control. The human brain evolved to optimize the outcome of our behaviors. And as such, behavior should be one relevant factor of investigation when we study human brain dynamics. So how then can we investigate natural human brain dynamics? This comes with different flavors. Usually what you need is some kind of a mobile brain imaging device, and that can be EEG, but can also be functional near-infrared spectroscopy. These are the two imaging modalities at the moment that would be mobile portable. And then, of course, from your group, intracranial recordings, which provide a way better signal-to-noise ratio in the recordings. They are less prone to volume, conducted errors, and artifacts. But the main point is that if you want to understand how the brain controls action and how action influences brain dynamics, then these two modalities have to be recorded synchronously. So you have to record motion capture to get to the behavior in parallel with EEG. And in our lab that you can see here in the background, this is one lab space dedicated to MoBI experiments where we have like 160 square meters that we can play around with in VR or in the real world. So this provides us obviously with a very nice control over the experimental protocol. And then one important modality that becomes more and more important to us, to our research lately, has been eye tracking that can be used in the real world using mobile eye trackers, but also with integrated eye tracking in the newer head-mounted virtual reality systems. If you do that, again, this comes with different flavors, and I took the liberty of showing your own setup. The general idea is the same. You need to combine motion capture with some form of brain imaging, and here again, combining that with head-mounted eye tracking or mobile eye tracking basically provides you additional information that can be used to feed back into the EEG to extract, for example, events from eye movement behaviors like blinks or fixations, to then parse the EEG signal and analyze the EEG signal according to these eye movements. If you record all these data streams, all these different modalities, they usually come with different sample rates, like virtual reality with a very low sample rate, eye tracking with higher sample rates up to 250, maybe sometimes 500 hertz, and then EEG that you can record with 500 hertz or 1 kilohertz or even higher. So all these different modalities need to be synchronized with different sample streams, and that is a non-trivial issue. There are different solutions out there. One solution that is open source and freely available to everybody is LSL, Lab Streaming Layer. That's a software architecture developed by Christian Cote that streams or basically synchronizes all streams that are based on a wireless protocol and provides a mother clock basically for synchronization online and offline. So you can use information in your experimental protocol, for example, location in space, to feed that back into the protocol and, for example, present something in your virtual reality dependent on where participants are located in space or which heading direction they assume at the moment. You can also take EEG information, extract that, and feed information back through virtual reality, for example, dependent on the user state and many other aspects. So the data synchronization is a non-trivial problem, but the data analysis is also non-trivial. Multi-modal data fusion and analysis on those fused data is, I don't think, yet an established procedure. So there are many ways how to approach these questions or your research questions. And one thing that we have been developing is extraction of events from behavior, may it be eye tracking, may it be movement itself, to feed that back to allow extraction of new events based on that behavior of participants to then parse your EEG signal. That is part of the bMoBile pipeline that we already published on bioRxiv, and that also basically uses data-driven analysis approaches, in this case ICA, to dissociate brain from non-brain activity in these MoBI recordings. And as you are well aware, movement leads to artifactual activity, or activity at least that has been considered traditionally as artifacts stemming from eye movements itself. If we talk about volume conducted EEG activity, not the better signal to noise ratio that you get with intracranial recordings, but if you have simple EEG, then you get volume conducted signals with facial muscles, neck muscle activity, eye movement activity, and of course mechanical artifacts from movement of electrodes when participants move in space. And if you look at volume conducted signals, like the example that you see here, you see that if you back project the left motor cortex. I don't know whether you can see my mouse, but the oscillating first source here that was simulated is the left motor cortex, and then you have the right parietal cortex. These are alpha activities slowed down to basically see what's going on. If you back project that through a realistic head model, so the forward solution is a realistic head model here from Zeynep, then you can see that recording EEG on the surface of that participant does not allow to dissociate the different sources of activity in the brain. And this is only brain activity. Now there would be non-volume conducted, but capacity conducted activity patterns coming from neck muscles, from facial muscles, and from eye movement. So these data has to be separated to make sense and to get a better insight and additional information from neuroanatomical structures that we could localize using an inverse solution that can help us to interpret the signal and what's going on. And unfortunately, the standard head models that we use are very simple models. We do not have the luxury to have individual scans, fMRI scans, or even diffusion tensor imaging of participants that we would like to have to create a realistic head model. But if we use a standard head model, these head models do not include all these additional physiological activity sources like facial muscles, eyes, and the neck muscles. So one approach would be to improve these models, either using individual head models or using improved standard head models like HeartMood. That's like the head artifact model using tripoles that we recently published. That includes facial muscles and all the other sources that will significantly contribute if you have actively behaving participants. With these kinds of data-driven analysis approaches, and independent component analysis would be one example of a blind source separation or spatial filter approach, you can dissociate brain from non-brain activity, and that now allows activity of participants in your experimental protocols, like you can see here in the left video. This is like one of the very first setups that we recorded. That was in 2007. We started with the recordings and then ended up in 2009 in Michigan at the lab of Dan Ferris, where we have like multiple modalities being synchronized here with EMG, motion capture, high density EEG, while participants were running, walking, and standing on the treadmill, working on this visual oddball paradigm. And the data is not really nice to see if participants are running on a treadmill. This is something that you don't really want to see if you record EEG. But the point again here is that using movement information, in this case, gait cycle artifacts that you can be used as to compute a template of channel-based gait-related artifacts to subtract that from the original signal and then run a blind source separation algorithm on the data, we were able to investigate brain dynamics even in participants running on the treadmill and doing this visual oddball task. And this is just one example, and the prototypical example of behavior being used to clean an EEG. So usually it's eye movement behaviors that are extracted from the data to clean that from the data, back project to the sensor level, and then analyze the data. But once you have the sources, you can also go ahead and basically start interpreting and analyzing the source activity. And this again is the example of the setup that we had back in Michigan. And on the right-hand side here under B and C, you can now see the results from independent component analysis on several subjects that we ran in this experiment. And what you can see here is that in yellow, there are tons of independent components, with the smaller spheres representing individual ICs and the bigger spheres representing centroids of clusters using k-means clustering of these ICs in multidimensional space. You can see that you get a lot of ICs that are located outside the brain volume, very much aligned with the skull. And if you look into the activity patterns, you can see that these reflect neck muscle activity. You do get, of course, eye movement related ICs here in gray, and you get brain ICs in different areas of the brain. which you can then further analyze as we did in these previous studies. What I would like to show you today is like a short journey of a few experiments, like 3 experiments, where we showed that brain dynamics differ when participants actively behave from what we know as traditional brain imaging studies using desktop setups. And I would like to provide insight in how we try to match behavior with brain dynamics, which is not very straightforward, as I would argue. So first of all, if you want to understand behavioral state changes and their impact on brain dynamics, then you should start with a paradigm that is well known and well published, so that reviewers would basically not be able to argue this is a mobile EEG. You can't analyze mobile EEG because it's too artifact laden. And we heard that for several years. So we started with an oddball paradigm, which usually is something like that. You stare at the screen, you stare at the fixation cross, and then every time there's a color change to a target color, you would press a button. And we took this paradigm, made it a little bit more realistic, and projected that on a big screen in front of participants, got rid of the fixation restrictions, and made it a dynamic moving paradigm to be a little bit more realistic, at least. So, and participants basically were asked to respond to color changes, to a color target here that was blue. Among distracted trials, change to green, and that you can see here. And we compared a standard desktop scenario where participants were simply standing in front of the screen and pressing a mouse button whenever the color changed to the target color. And we compared that to a physical pointing condition, which you just saw in the example, where participants would intercept the trajectory of the moving sphere to touch it on the screen. If you look at the response times, you can see in red the physical pointing, so the MoBI condition, and in blue you can see the button press condition. And for the physical pointing condition, we defined the onset of the response as 10% of the maximum velocity of the movement towards the screen. And what you can see here is that using this kind of a MoBI scenario, participants were significantly faster. They were still faster if you used a cutoff of 40% of the maximum velocity. So there is, or there seems to be, a quicker response in this kind of setup, and this is quite also reflecting the motivation that participants had in this kind of setup. They had simply more fun interacting with the sphere rather than just clicking the mouse button. If you look at the event-related potential, so this was like in time point 0 here indicates the moment of the color change at electrode PZ or close to PZ, so that's a custom montage that we're using here. And this is one of the electrodes where you would usually look for a P3, an oddball P3 or late positive complex in the ERT. And this is what we did here. You can see in black the standard trials, and the solid lines are the pointing, the physical movement condition, and the dotted lines are the button click, so the mouse click conditions. In red, we had the distractor, and in blue, you see the target trials. And what you can see is that for the target trials, you do have late positive complex, which is much pronounced and significantly more pronounced than the distractor trials or the standard trials. And what you can also see is that for the onset of the target in the pointing condition, there's a massively increased amplitude in this late positive complex. If you look at the timing, this is starting around 400 milliseconds. And if you go back to the response times, what you can see is that this is approximately also the time where they start responding to the target. So the question was, is that a simple artifact from a sudden movement onset that would basically lead to a contraction of the neck muscle to stabilize the head in space, and which might be associated with a strong artifact in the sensor recorded signal? So we ran independent component analysis. And if you have questions to that, we can discuss that anytime. And if you have questions, just ask, please. And so we used ICA. And what you can see here again is in gray, independent components reflecting neck muscle activity. The bigger spheres again reflect the centroids of clusters using K-means clustering. The white clusters here represent eye movements, and then the colored clusters represent brain sources. And we focused on the parietal cortex, and we found 2 clusters. And what you can see is a wide spread of independent components. Again, this is EEG. I would not claim that we have like a very precise localization in the parietal cortex here. There's a massive spread of single ICs. And this is one of the issues that we have with EEG and standard head models. But if you use those clusters and back project to the sensor level, so basically what you do is you use a butterfly plot, you plot all channels over time on top of each other, and then you take the minima and maxima at every single sample point, and you create an envelope of the surface sensor data, only back projecting brain sources. And if you now look at the back projection of the 2 parietal clusters to this envelope, in black you can see the envelope, and in red you can see the contribution of both clusters back projected to the sensor level. And here the gray area, that is the area where we analyze the data, the late positive complex. Here on the left column, you see the key press condition for the standard stimuli. So these are the trials where the standard would appear and participants would press a key. And for the standards, they wouldn't have to press a key. And this is the pointing condition, so the MoBI condition, where you see a slightly increased envelope, slightly increased parietal activity, but that is not significant. So there was no difference between the 2 conditions. And then for the targets, what you can see is a nice increase, a late positive complex, as you can see here. And you see a significantly increased activity of the 2 parietal sources contributing to that envelope. And the question now was whether the huge P3 amplitude or late positive complex that we saw on the sensor level would simply be an artifact that now would disappear if it was related to muscle activity. But this was not the case. If you back project only brain sources and get rid of the traditionally considered artifacts in this recording, then you can still see a significantly increased late positive complex in the pointing condition as compared to the target, to the key press condition. And you can also see a significantly increased contribution of the parietal cortex to that envelope. And that is not surprising, since this is an embodied process, right? It's simply computationally more expensive to continuously compute the future position of a dynamically moving object in space and program potential motor response to intercept the trajectory of that moving object. So this is computationally simply more demanding. And this is an example for embodied cognition. And this is one of the reasons why I think recording behavior and brain dynamics will provide us new insights and what the brain is able to, or basically controls how the brain controls movement. So this was one of the first studies that we used to investigate the relation between brain dynamics and active behavior. And over the time, we became more complex. We were more confident that we can actually record brain dynamics in actively behaving participants and that we are able to analyze the data using simple EEG. And we dove a little bit more into spatial cognition domain, and this is what I'm going to show you now. So this is an experiment where I tried to really get to the core of movement, active behavior, and how that influences brain dynamics with respect to heading computation. And we all know that in our natural environment, we navigate all day long, we rotate, and we have tons of sensory input contributing to updating of orientation, position, and heading in space. And that includes visual information, vestibular information, proprioceptive information, all the kinds of information, of sensory information, idiothetic information that we do not have when lying supine in a scanner or sitting on a chair in a dark experimental room. So this is a setup before the lab got refurbed, and you can see a head-mounted virtual reality. That's the old Oculus system. The backpack where we have a Zotac gaming computer, and then we added motion capture. This is like the face-based system. That's an active LED system, camera-based motion capture system, where we added like heading information on top of the heading information that we get from the VR system and torso. And so basically the task here was very simple. Participants stood in the middle of the lab, and this is a bird's eye view of the setup, and then they would see an orienting beacon that they would have to align to, and that was then replaced, initiating the trial by a sphere that would start moving at a defined distance with different velocity profiles to end unpredictably on the trial at an end position between 30 and 150 degrees eccentricities. We had a few filler trials, and obviously we did that to the left and to the right. And after the sphere changed its color, that was the imperative stimulus for participants to rotate back and indicate the initial heading at the beginning of the trial. And with that, we get all the behavioral measures that we need: over and underestimation, duration of outward and backward rotation, and all the things we were interested in. And we looked at the accuracy of the back rotation dependent on the eccentricity of the outward rotation. And we clustered the outward rotations here just for gaining like a little bit more statistical power. And importantly, again, like in the previous study, we compared a traditional desktop setup where participants were standing in front of a huge screen that you can see on the right side here in the video, and controlled the rotation using a joystick. The visual input was more or less identical to what participants saw in the mobile brain-body imaging condition on the left side, where they were able to actively, physically rotate. And in this condition, you get all the idiothetic information that you would have in natural movement through the environment. So proprioception, vestibular information, visual information. While on the right-hand side, in the traditional setup, you only have the visual information and some kind of a translational proprioceptive input through joystick movement. If you look at the behavior, so what you can see here is the mean absolute error as a function of the eccentricity or the end position of the outward rotation. And in blue, you can see the stationary, the 2D condition, basically. And what you can see is more or less a linear increase in absolute error with increasing eccentricity of the end position. And for the mobile 3D condition, there are like two interesting points. First of all, there's a main effect of the movement condition. So if you actively rotate, the absolute heading error is significantly reduced by half nearly. And then there is a main effect of eccentricity of end position with an increasing absolute error with increasing eccentricity, but there's also an interaction. And this was as you can see, there's not much of an increase in the absolute error up to 90 to 100 degrees. And after that, the error and the reproduction of heading error increases. And this is basically when you start crossing your cardinal body axis. And there seems to be a switch in the spatial representation that is used to basically recompute your initial heading once you rotate over your cardinal body axis, which is reflected in the increase only after the 90 to 100 degrees. Excuse me. So the question, however, was, do we get any kind of neural signature that might reflect vestibular information? So acceleration or velocity-dependent manipulation of brain dynamics that would indicate that they use vestibular information to update orientation or heading in space. And we've been working around this for quite some time. And in the end, we came up with a representational dissimilarity analysis. And basically what we did here, so I walk you through that, is using an example of neck muscle activities. So what you can see here is a cluster of neck muscles that will contribute, of course, to the head rotation in the active physical rotation. So we would expect some kind of neck muscle activity when participants actively orientate and rotate in space, as compared to the condition where they would stand in front of the television screen and just use a joystick to rotate. So there's no head movement in that condition. So we used these clusters, and this is the example cluster, and we basically computed the velocity profile of the outward rotation, and we used a relative velocity binning from the lowest to the fastest velocities and bin that in 10% bins, and then sorted these short epochs according to the velocity. And we also used the epochs brain dynamics, so the power in different frequency bands for the different frequency bands, as you can see here, from 3 to 30 Hertz. And we sorted them according to the velocity in these epochs. So what you can see in these kind of arrangement here in this figure is the sorting of power, in this case, theta power, for the lowest velocity, 10% velocity epochs to the fastest 10% velocity epochs. And if there is some kind of a velocity dependent modulation of brain activity or power in different frequency bands, in this case, you should see this kind of a striping pattern, right? Here you can see that the lowest velocities lead to a desynchronization or less synchronization in theta as compared to the faster rotations. And we did that on the left-hand side, of course, for the physical rotation and the joystick rotation. So this was our first step. And then we basically went ahead and computed the dissimilarities between all these different velocities and all different frequency bands with each other. And this gives you like these 180 times 180 dissimilarity matrix, where you have the active rotation condition in this upper left triangle. Then you have the direct comparison of the physical rotation with the joystick rotation. And then on the lower right triangle, you see the dissimilarity matrix for the joystick condition only. And then obviously you do a permutation statistic on that to get the statistical differences. And overall, this gives you a number of parameters to look at to investigate whether and where brain dynamics would impact, behavior would impact brain dynamics. And if you look at this neck muscle cluster, the right neck muscle in this case here, you can see that there is a velocity-dependent modulation of different frequency bands, basically a wide range of frequency bands, more or less only in the physical rotation condition, but much less pronounced in the joystick rotation condition. And if you look at the dissimilarity matrix, you can see that this is reflected in dissimilarities across different frequency bands and also across different velocities, and more pronounced so in the higher frequency range if you now compare in the lower square here, the active rotation with the joystick rotation. And what was important for us here, that is the sanity check. If you look at the joystick rotation, there's no velocity dependent modulation of neck muscle activity. And that makes sense. You see that here in the permutation tested, there's no significant change dependent on velocity in this neck muscle cluster, because in the joystick condition, participants were not moving their head. So it's static condition, and the visual flow only basically gives you the rate of velocity here. So this was a nice sanity check that told us, okay, our method might work actually. So we went ahead and looked into some of the areas that were of more importance to us. For example, the retrosplenial complex, which is the hub to translate between egocentric information coming from multisensory input into an allocentric representation that is housed in the medial temporal lobe, as you all know. So the retrosplenius can be used to coordinate that egocentric information using external landmark information, for example, to translate that egocentric into an allocentric reference frame. And what you can see here is that using the same approach, there is activity in both conditions. They differ, and there is significant difference specifically in the lower frequency range for the physical rotation as compared to the joystick rotation. But you do see RSC activity also during joystick computations of heading. And that replicates a number of studies with EEG of our lab, but also many fMRI studies showing that the RSC is active during these navigation tasks in a number of scenarios. So visual information is sufficient to update heading. You don't need the entire range of sensory modalities to update heading, but obviously there is a relation of accuracy in heading computation dependent on the number of modalities you have during the rotation. Another very interesting example is the occipital cortex. And what you can see here is mainly the inverse pattern compared to the neck muscle cluster I showed in the beginning. What you see is there is not much of a velocity-dependent modulation in different frequencies during physical rotation, but there is velocity-dependent modulation of a wide frequency range during the joystick condition. If you look at the dissimilarity matrix and the statistical test, what you can see is that the two movement conditions differ massively in the low frequency range up to the lower beta range, mostly in the low frequency range up to alpha. For the physical rotation, there is no difference. There's no velocity dependent modulation in occipital cortex, but there is the massive difference mainly in the alpha range and the lower alpha range here for the for the joystick rotation. And this replicates what we know, the alpha desynchronization that we see in traditional desktop scenarios of spatial orientation, heading changes, navigation, that was usually interpreted as being indicative of cognitive processing and resources being, used in posterior brain regions, which in this case seem to be more potentially processing of sensory mismatch between the visual input and the other idiothetic senses. Like there's no vestibular information or proprioceptive information about the rotation, but only visual information. So this might be aligned with predictive processing or predictive coding, basically, of what happens when you actively move in the environment. I'm going to jump over that. This is simply a visualization of a single trial. This is time-warped event-related spectral perturbation in the joystick rotation condition with the alpha desynchronization that I was just talking about, as compared to a physical joystick, physical rotation, where you can see no such alpha desynchronization, but a massive increase in the theta frequency range, and the differences between the two conditions is massive. So this again supports what I just showed in the dissimilarity matrix, that the lower frequency during the rotation only in the active physical condition massively deviates from what we know from traditional desktop setups. And I would like to show you one last example of multi-sensory contribution to spatial learning, staying only on the behavioral level. This is a setup where we are working together with the Charité, investigating right hippocampal resectomy patients. So we have 10 patients with the entire right hippocampus removed. And you can see here on the right side an example of a virtual Morris water maze, where we have 3 landmarks and participants basically trying to find treasure locations using landmark information. And this is the example of the MoBI setup, where you can see participants moving around in the maze. And again, we did that in a stationary condition and compared that with a full mobile condition. This is a mobile condition where participants learn the location of an object in 3 consecutive trials. So they try to find the hidden treasure, and then basically using the landmarks to remember the location of these treasures. And we repeat that 3 times, always starting from the same location. And then we basically go into 4 probe trials, where we now rotate the starting location. And one starting location will be the same as during the learning trials, but 3 locations will be rotated. And this is the probe trial example where participants will move, locate the flag where they think the target was located, then confirm that, and the next probe trial will start. As I said, we are going to compare that with the desktop scenario where participants are standing in front of a screen, like we did that in the spot rotation experiment I just talked about. We had 2 scenes alternating, balanced across trials. And again, here's an example of one patient. I'm going to show only the probe trials. So this first probe trial that you can see here is a same probe trial starting location from the previous learning trial. And once you rotate the starting location, what you can see here is a simple replication in the desktop scenario. So standing in front of the screen, of a path that they remembered that would lead to the location. And this is done irrespective of the landmarks in the desktop condition. So there's no adaptation, there's no use of allocentric information, but simply a replication of an egocentric, visually stored trajectory that led to the trial. In contrast, if you look at a movement in the mobile brain-body imaging setup, you can see here again, same starting location in the first probe trial. But then what happens is that patients in the MoBI setup use different strategies. And obviously they, in this case, move first to the center of the lab, reorient using landmark information, and then localize and find the treasure pretty accurately, actually. And they do that in basically all rotated conditions. So if you plot the presence probability, and this is now basically rotating all starting locations and end locations across participants to one location. So you see that in the desktop, the presence probability is more or less distributed without a clear pattern. But in the MoBI condition, you can see the starting locations. You can see that there is a higher presence probability in these locations, indicating that participants actively use landmark information, so they stand longer in their starting locations, and then they are more likely to end up in the goal location. In this case here, rotated all conditions to this one location. So this indicates a different strategy when you have all idiothetic information. And this is reflected in a number of navigation efficiency measures. If you look at temporal efficiency, and we're looking at learning trials here in this slide, you can see that the time in seconds to reach the goal in the stationary as compared to the mobile condition. In blue, you see the patients, MTLR with the medial temporal lobe resectomy, and the control patient, control subjects in orange. You can see that in the desktop condition, patients take more time to find the location, they are less precise, and as controls, and they improve significantly when they have all idiothetic information. This is also the case for spatial efficiency measures, like the path error to the final location or the surface coverage. If you have all multimodal information, the surface you cover reduces significantly. This is the case for both patients and controls, but more so usually for the patients. And these are the probe trials. And our patients here in this study are very good, actually. So they are also in the desktop condition already very good and do not show the expected pattern of being less efficient temporally or spatially as compared to controls. So they are very good from the beginning. But the point is that both patients and controls significantly improve with presence of all idiothetic information. And if you look at spatial memory performance in the probe trials, you can see that the memory score, this is simply the percentage of trials if you distribute random dots in the arena, and then you basically select random dots, compare the distance of the random selected dot with the actual indicated dot by the participants, then you compute the number of dots that would be further apart than the indicated location, then you get the memory score. So this is a percentage score here of number of dots being further apart than the indicated location. You can see that now you find this interaction of patients as well as controls improving significantly in the MoBI condition, but patients having much more benefit of the presence of all idiothetic information as compared to the controls. And if you look at the scatter of the final location, this is only the distance between the finally indicated target locations. This is more or less the same pattern with a significant improvement for both groups, but much more benefit for patients in the full MoBI condition. Okay, with that, yeah, I basically would summarize what we have found, and that is what you also have found in many experiments thus far, is that if you actively move, by definition, movement will lead to sensory information that is processed by the brain, and only that will change brain dynamics. So it's very simple and straightforward that movement or the behavioral state will change the brain dynamic state. But that is a very basal assumption here. What we obviously are after are higher cognitive processes and the associated neural dynamics that will change dependent on the movement. And I hope that I was able to show you how we try to address that. If you now move into the real world, and this is obviously one of the ultimate goals, to have like continuous recording of brain dynamics in participants actively behaving in the natural environment 24/7. This is something that we also start addressing. Now it becomes a little bit more complicated because you have no control over experimental factors. I think like in LA, in Berlin, nobody really cares whether you run around with a colorful cap and with the EEG on top. So it is possible, and people do not, are not disturbed. It's not too intrusive using EEG in the real world. But with that, you will add an additional layer of potential artifacts, non-controlled factors that will influence your brain dynamics. So there are several issues that have to be addressed in my perspective in the future. In summary, first of all, I think more than a decade of MoBI studies in more and more labs, and this is a really fastly developing field, have demonstrated that the behavioral state changes the brain dynamic state. And that is an important insight because we now get closer to an ecologically valid explanation of brain function. That idiothetic information that stems from movement changes brain dynamics in diverse brain areas as compared to traditional desktop protocols, and that MoBI allows for new insights using that kind of approach into human brain dynamics, accompanying natural embodied cognitive processes. And that can be in VR, can be in the real world. I don't think that there will be much of a difference if you have a good virtual reality environment to work with. But I think we are far from where we want to be, actually. And there are a few challenges that will have to be addressed for us to really make progress in this field. And I think these next challenges include the development of new hardware, providing continuous access to whole brain activity. And I think your lab, Nathania, is like a lead here. So this is unprecedented data that you produce and that we see that contributes significantly and shows what is possible. I think what you also address is that you overcome scale chauvinism, right? So that single neuron local field potential, macro scale, EEG activity, that these different research areas usually did not talk with each other. So there's an ignorance of the results from different fields, which has to be overcome. And we should be open to address as many modalities as possible. FNIRs on top of it, peripheral physiology. I've been talking to some of you before. So we need a more holistic idea of what is going on to better understand how the brain realizes or controls these complex processes. And I think that the combination of such recordings with variable multimodal physiological recordings will propel us to new insights, and including eye tracking will be one important aspect, motion capture and other modalities, and the question, how can we get that into the real world, and how can we potentially record all this data for longer durations, longer time periods, potentially using cloud-based online processing of the data and storage over years to develop models, individual models of brain function dependent on this kind of complex information. And I think one of the biggest problems that we face is that we need smart and new analysis approaches to address very low number of trials and events that we have in these kind of scenarios with the complex contextual information, where we run into the issues of overfitting our models when we try to get our results out of it. So we need new analysis approaches, and I think this is one of the most important issues that we have to address in the near future. And with that, I would like to thank everybody who contributed to the data that I've shown here. And if you're interested, please all be aware that next year, 2024, we have the next Mobile Brain Body Imaging Conference in Slovenia in June 2 to 5, and I hope to see many of you there. Thank you very much.
Thank you, Klaus. Fantastic work. We have any questions from the audience? You can put them in the chat, please, or just raise your hand and unmute. Okay, question, Suleiman.
Hello, I have a question about some of the early studies that you showed, where you adapted the desktop version to the in-person version with the bigger screen with the moving target. I don't exactly remember the name of the task, but so when you were comparing desktop versus in-person, but then I noticed that there was also a difference of the moving target. So in the desktop version, was the target also moving, or was it just static?
So you're talking about the moving sphere on the screen?
Yes.
Yeah, no, that was the same. So it was the same screen that participants stood in front of, and they, in the stationary condition, they simply had a mouse in their hand. They were just, standing in front of it and would push a button whenever the target appeared. So it was the exact same visual input, no difference in the visual input, but the response was different.
Interesting.
Okay, I see. Perfect. Thanks so much.
Matthias?
Hey, again, thank you so much. This was really, really fascinating. I'm just really curious about the patient data that you get. This is really fascinating, but I'm trying to make sense out of this result where you find that, people do much better in the physical condition, where basically they find the target locations in the physical condition, but not in the non-walking condition. Do you have any, maybe it's just speculation, I don't know, but do you have any insights in what's going on? Do you think they use a different strategy when they have all kind of the sensory cues? Or I'm just trying to find an explanation for why this is possible. Do you have any insights?
Yes. So first of all, I have to qualify that a little bit, because they do have an entire left hippocampus, right? So there's obviously, to a certain degree, a compensation of what's going on here. But yes, our point is that we're talking about extra hippocampal information or strategies, basically, to code location and memorize location. And we are currently analyzing the signal. We are focusing on the parietal cortex and the retrosplenial complex. And we do expect differences in these areas. It's a little bit difficult, actually, as you all know, to analyze the data, and we are right in the beginning, so I can't really give you any solid answer to this. One of the issues, however, or one of the parameters that we are trying to match with the brain dynamics during the analysis is IDFI, or basically the accumulated head movements in the beginning of these trials. And there is a difference in the trials, and you saw that in the presence probability, that when participants actively move, also the patients actively move into the space, they do use different cues. They do use allocentric cues. And this is, I don't know, it's a question how successful they use that, or whether the example that I showed, right, is if you move to the center of the lab and then you basically use a landmark as a beacon to navigate to the landmark, because you localize or memorize the treasure next to that landmark, then it's kind of a beacon navigation that you do. And that might explain this specific pattern, just moving into the center of the lab, using a landmark that you previously localized, and then return to the treasure. So this might be one of the strategies happening here.
Really fascinating. Yeah, I was asking, I saw a kind of similar effect also in a paper from Della and Lucas Kuntz, where I think people also changed their strategy when they were like at risk for developing Alzheimer's, which is kind of a, anyways, it looked similar, but it's really, really fascinating, and but still hard to understand exactly what's going on. Anyways, thank you so much.
Yeah, you're welcome. I'm super curious about the data, and also I talked to Nathania about that. I mean, I would like to have forward models, but the question is, how do you model a forward model with a huge hole in the medial temporal lobe? So lots of questions that we have for the source reconstruction here too. So Lehman?
I had a follow-up question to my previous question. I think in that task, you found that the reaction time was faster or shorter for actually touching the visual stimulus. I was wondering why, because instinctively, I think that just clicking the button would take less time than going and touching the screen. So I was wondering why you think the reaction time is shorter.
So I have difficulties understanding you. So you're again at the moving sphere experiment?
Yeah, so I think you had a
yeah.
figure showing the reaction time was faster when a subject was touching the screen.
Yes, indeed. They were significantly faster. Even if I said like up to 40% of the maximum velocity in that direction of the movement, we actually explained that by participants being simply more motivated in this task. They all the participants in the end, in the exit interviews, basically said they had fun actually doing this kind of experiments, a little bit like whack-a-mole, right? So it's more interesting than our standard protocols where you sit or stand in front of a screen for hours and then you press a button whenever something happens that you're instructed to do. So they had more fun, and I think this is motivated behavior, and I think that's something that we come to address more and more in all our VR setups, because people are much more involved. They have fun actually participating in our experiments when they can explore virtual worlds and things like that. This is much different than everything that we did for decades, where we had participants simply sitting in front of small screens and, you know, pushing a button.
Yeah, I think thank you so much. Anecdotally, that's our experience as well, but it's really nice to see some data, you know, showing this, what we see in our participants here. So is it what I understood correct that you hypothesized in the last study you presented that you may see some changes in the brain in sort of compensatory extra medial temporal areas to compensate for this difference in behavior?
So we're not yet at the brain dynamics, but yes, the behavioral dynamics. So like IDFI, like this is one indicator that clearly differs between groups and between movement conditions. And then you see this strategy change. I think like the example that I showed of this one subject N, where she really just replicates a visual trajectory that she encoded in the desktop condition. And there's no adaptation. So there's absolutely no use of landmarks at all in this desktop scenario, while being mobile allowed her obviously to use a different strategy. So, and there is variation between patients, massive. So this is going to be difficult to address. But yeah, I'm very curious about the EEG data, but I unfortunately can't say anything yet.
Very interesting. Any other questions? I think we're out of time, so maybe that's where we'll end. Thank you so much. Super fascinating work. Just really relevant, especially for our work. So I'm glad the trainees also could hear some of the history. And thank you so much, Klaus, again for
Thank you
joining.
very much for the invitation. It was my pleasure. Okay, hope to see you soon.